Hello and welcome to PrESSTo. PrESSTo, the Promoter and Enhancer Slider Selector tool, is a web tool and database of promoters and enhancers obtained during the FANTOM 5 project. At the apex of transcriptional regulation lies the transcription start site and the core promoter around it. At the TSS core promoter region, all the signals from enhancers, transcription factors, insulators, etc. are integrated and the decision to start transcription is taken. The FANTOM project set out to find all the promoters and their usage across the human body. To do this, they developed a method called CAGE, Cap Analysis of Gene Expression. CAGE captures capped mRNAs and only sequences the first 20 to 30 base pairs of the resulting full-length cDNA. The resulting CAGE tags are mapped back to the human genome, resulting in peaks at active transcription start sites. The number of tags correspond to the expression level, much like in RNA-seq. So we get both the location of promoters and expression level for the cell we perform CAGE on. The FANTOM project applied CAGE on a multitude of human cell types, tissues, and cell lines to create a comprehensive atlas of promoters and their usage across the human body. Here we see an example of CAGE analysis around the annotated start site of the GALT1 gene. Each bar plot shows the CAGE tag intensity for every nucleotide for a set of cells: T cells, monocytes, aortic smooth muscle cells, and SW13 cells. Here we see that there are at least two different larger start site clusters which have different cell usage preferences. The T and SW13 use the left ones, the aortic smooth muscle cells use the right one, and monocytes use both. So even a single gene may have multiple promoters for different cell usage, and depending on the cell of interest, different promoters should be selected for further analysis. When examining expression data like CAGE at transcription start sites, we only see the effect of regulation, not the underlying mechanism. What if we could see the enhancers as well? Here we observe the known enhancer from the VISTA database. It has all the features we have come to expect from enhancers. It is highly conserved, DNA sensitive, has H3K27 acetylation and H3K4 methylation marks, but not H3K4 methylation 3. But it also has summary chip transcription factor peaks, which we had to squeeze together in the image. In addition, we can also observe two CAGE peaks that delineate a narrow region right in the middle, just where the conservation is the highest. These are bidirectional enhancer RNAs. In FANTOM 5, we used the mentioned properties to predict enhancers based on CAGE data. This resulted in around 43,000 enhancers. We found that the expression levels of enhancers corresponded to their usage in cells. So much in the same way as the promoters, we could use the FANTOM 5 collection of cell types and tissues to create a unique enhancer usage atlas across the human body. Most users of the promoter and enhancer atlases would like to select a subset of enhancers and promoters which are only used in the cell or cells of interest. However, most promoters and enhancers are not exclusively expressed in one cell type. We find that it is more relevant to use constraints like at least 30% of the total expression from this promoter or enhancers should come from cell type X. In PrESSTo, we have taken this concept one step further to let users interact with sliders to create expression rules to select enhancers or promoters. I will now show you some user scenarios for both enhancer and promoters using PrESSTo.